Hello and welcome back to Teach Me in 10. In this episode, we're joined by Dr. Nina Jajcanin Jozic, Senior Application Scientist at Refeyn. We asked Nina how Refeyn is changing the game in therapeutics development, and she'll reveal how a groundbreaking technique called mass photometry is helping researchers unravel the complexity of next-generation antibody modalities. Hi Nina, how are you doing?
I'm fine, thank you. How are you?
Good, great. Thanks for joining us today. So we're chatting about sort of the challenges and innovations in biologics development with a focus on next generation antibody modalities. So to set the scene, what trends do you see in antibody therapeutics development at the moment?
So, therapeutic antibodies are one of the largest class of biotherapeutics. And for a long time, the focus was on monoclonal antibodies. And these are antibodies that are made to target and bind single specific antigen. And monoclonal antibodies are sort of considered as a traditional therapeutic antibodies. But what we see lately in antibody therapeutics is shift with the focus moving from these conventional monoclonal antibodies toward the new modalities. And these new modalities are pushing boundaries for efficacy, safety, and resistance mitigation.
Great, that's really interesting to see how the field is evolving. So with all these new approaches emerging, what are the new modalities?
So there are a few. Anything from IgG fusion proteins, so fusion protein of the regular antibody, FC engineered antibodies, antibody fragments, and nanobodies, which are quite interesting and they're tailored specifically for challenging targets like ion channels. But the focus really is primarily on two types of these new modalities. So we have multi-specific antibodies, to be more precise, bispecific and trispecific antibodies. Now, these are the antibodies that bind not only one, but two or three different antigens. So they have multiple targets. And the second type is when you basically have monoclonal antibody and an additional structure has been added on this monoclonal antibody to make it more efficient. So then we have ADCs, which are antibody drug conjugates. Now these can carry cytotoxic payloads. Then you have radioligand antibodies. And then you also can have antibody oligoconjugates. And these can carry, for example, siRNAs for gene silencing.
Wow, so quite a shift now from traditional antibodies.
Quite significantly, yeah.
And I imagine these more complex formats must present some unique challenges. What are the analytical challenges associated with these modalities?
The major challenges would be assessing stability and developability. So you need to check all of these novel antibodies. You need to check how their stability looks like under stress, force degradation, different pH, different temperature. You need to be able to detect the high order structures like aggregates. You need to be able to detect if these antibodies are fragmented. And to do that, you need to have a very precise analytics. And then speaking, for example, about multiple binding antibodies like bispecific, trispecific, you really want to assess function and affinity. So because you have a multiple binding sites, you need to validate each of those binding sites because it will target different antigen. So then that reaction, that affinity is going to be different. So you always need to make sure that yes, it's binding to a right target, and yes, it's binding in the right stoichiometry. But I would also like to sort of point out that with these novel antibodies, with these new modalities, we see more and more of them. And it's getting more and more complex, which is really great because their efficiency is much higher. But then it takes a long time to develop these. So really challenges processing time. It's the complexity of the technology that we are using because there's many technologies that are used for assessing critical quality attributes of these antibodies. So that takes time, that takes expertise, and also you use a lot of samples. So the sample consumption is really one of the issues. So I would say that also presents a huge challenge in the field.
So it sounds like traditional methods aren't quite cutting it anymore. So what are newer technologies that are overcoming these challenges?
So clearly, the industry is responding with high resolution and orthogonal technologies. For example, mass spectrometry, liquid chromatography, and charge-detected mass spectrometry technology is quite well developed. Microfluidic immunoassays, high throughput antigen screening, and of course, mass photometry.
Yeah, so you're a senior application scientist with Refeyn, who's the pioneer of one approach in particular. Can you explain what mass photometry is?
So, besides the fact that this is a really cool technology, mass photometry, putting it very simple, measures the mass of molecules with light. Now, to go into more details, how is it done? You will have your molecule of interest, let's say it's an antibody, in the solution, and then you will position your sample on the cover slip. And that cover slip is positioned on top of the objective with a laser source. That light will go to your sample. So some of the light is going to be reflected on that surface, but some of the light is going to go through and then hit your molecules. And those molecules naturally will scatter that light. So what happens is that this scattered light basically interferes with reflected light. And this is what we measure. We measure this interference, and the result is ratiometric contrast. And this ratiometric contrast is directly proportional to the mass of your sample. So that means that you can measure with high precision and accuracy anything from 30 kilodaltons to 5 megadaltons when it comes to proteins, or even 6 megadaltons when it comes to AVs, for example. The measurement is done in 1 minute with nanogram amounts of sample and with technology that almost needs no development. So it's very, very simple and user-friendly.
Well, that sounds like a really elegant technique. Now, I'm curious about the practical applications. How is mass photometry applied for antibody analysis?
It can be applied all the way from discovery to development. And you remember we were talking about those analytical challenges. And this is where mass photometry really, can contribute. So, for example, if we look at the earlier R&D stages, mass photometry can really nicely complement the binding studies. Mass photometry can really give you nice information and precise information on reagent quality. Do you have expected mass of your antibody? Is your antigen of desired purity? And then also you can quantitatively measure antigen-antibody interactions, and you can determine complex stoichiometry when you have multi-component mixtures like, trispecific antibody with the 3 different antigens. You can also determine the dissociation constant and affinity metrics in real time. When we talk about later stages in development, you can, again, going back to analytical challenges, you can very precisely and accurately assess, for example, aggregation when you are comparing, let's say, different buffer compositions. You can assess aggregation, fragmentation in force degradation studies and, let's say, accelerated stability studies. You can detect very low abundant species, let's say up to 0.5% of aggregates of the total sample. And then what is really important, you can resolve these different aggregate species, so dimer, trimer, pentamer, hexamer, because the resolution of mass photometry is really, really high. So you can resolve these with baseline resolution.
Great, thank you. And I'd love to dive deeper into a specific example. Can you give us more details on your most recent work with bispecific antibody analytics and mass photometry?
Yes, sure. So recently there's been a lot of hype about a new kit on the block, which is antibody called Ivonescimab. So this antibody has been shown to be highly efficient in latest clinical studies. And this antibody binds 2 targets, VEGF and PD-1. Now, the ongoing hypothesis is what researchers have seen is that when ivonescimab is incubated with VEGF, there are these high oligomeric structures formed. And the current technologies didn't have enough of the resolution to really deep dive and understand what type of these high order structures were. So we decided to do that with mass photometry. The other reason why we were interested is that because with mass photometry, we would be working with the concentrations that are closer to physiological concentrations. So that also is quite important. And what we actually could see, we could resolve every type of these Ivonescimab VEGF complexes. We could show that there is only one predominant structure, and it's actually is not one of the highest order structure. It's actually one of the smaller ones. And then it was really nice because we could calculate the affinity, the association constants for each one of these complexes.
Amazing. Thank you for sharing those details. Sounds like amazing work. So lastly, we're just running out of time now, but looking ahead, what are you most excited about for the potential future applications of mass photometry for the antibodies field?
Yes, so clearly expanding analytics for these novel formats, multispecifics, and next generation new modalities, antibodies. I would like to finish with something really interesting that I've heard just recently, and it was the iceberg analogy, where somebody suggests that we should think about new modalities like the iceberg, and then current analytics show just the tip of it. And we really, are looking at advancing the next generation analytics that is crucial for developing next generation therapeutics. And we already showed, and I strongly believe that mass photometry is going to have a huge role in this area.
Amazing. Sounds like an exciting future ahead. Nina, thank you so much for joining us today. It's been really great talking to you.
Oh, thank you for having me. This was quite exciting.
Thank you. Take care. Thank you for joining us for this episode of Teach Me in 10, brought to you by Refeyn, who are pioneers in mass photometry, a revolutionary technique that enables rapid quantitative analysis of biomolecules at a single particle level. To learn more about how Refeyn is transforming biotherapeutic development, check out the links in the description, including resources on mass photometry and its applications in antibody analytics. If you enjoyed this episode, don't forget to like, subscribe, and stay tuned for more 10-minute insights from innovators driving the future of life sciences. See you next time.